Hello and welcome, or welcome back to another episode of Unicate Talks. In this series, we explore how scientific research is translated into real-world solutions that can improve health, well-being, and quality of life. Today's episode focuses on an innovation that sounds simple on the surface, but is grounded in sophisticated science, using a lollipop to help detect cancer. This episode has been developed with the consultation of a chartered scientist, Dr. Alando Watkis, whose insight supports the scientific framing, clarity, and wider significance of today's discussion. At first glance, the idea of a lollipop detecting cancer may sound surprising, even lighthearted, but this research addresses a very serious problem in healthcare, and it does so in a way that challenges how we traditionally think about diagnosis. To understand why this matters, we need to begin with the challenge it aims to solve. Cancers of the mouth and throat are often detected late. Early symptoms can be subtle, easily dismissed, or mistaken for less serious conditions. By the time people seek medical attention, the disease may already be advanced. Late diagnosis is associated with more aggressive treatment, reduced quality of life, and poorer outcomes. Existing diagnostic pathways can also be invasive and intimidating. They may involve uncomfortable examinations, biopsies, or specialist imaging procedures. For some people, fear, anxiety, or difficulty accessing specialist care can delay testing even further. The research explored in today's episode asks a different question. What if early cancer detection could be simple, non-invasive, and familiar? The proposed solution is a lollipop designed not as a sweet, but as a diagnostic device. When a person sucks on the lollipop, it collects saliva in a controlled way. Saliva is an important biological fluid. It contains thousands of proteins released by cells in the mouth and throat. Some of these proteins can act as biomarkers, meaning they provide early signals that something is wrong at a cellular level. At the center of the lollipop is a specially engineered material known as a smart hydrogel. This is not an ordinary absorbent material. It is designed to selectively capture and hold onto specific proteins while saliva passes through it. As the lollipop is used, saliva enters the hydrogel. Proteins within the saliva become physically trapped inside the structure of the gel. Crucially, the gel is designed to favor the capture of proteins that are of diagnostic interest, while allowing many other molecules to pass through. Once the saliva sample has been collected, the lollipop is removed and prepared for analysis. What makes this technology particularly innovative is how the captured proteins are released. The hydrogel responds to a specific wavelength of light. When exposed to this light, the structure of the gel changes, allowing the trapped proteins to be released in a controlled way. These proteins can then be analyzed to look for patterns associated with early-stage cancer. This light-activated release mechanism is a key part of the technology. It allows scientists to control when and how the proteins are extracted, improving the reliability of the test. At present, this capture and release process can take many hours. That timescale is far too long for routine clinical use. As a result, a major focus of the research is on speeding up this process. The long-term aim is to reduce the time needed to capture, release, and analyze the proteins, so that results could be available in minutes rather than hours. Ideally, the entire test could be completed in under 10 minutes. If achieved, this would allow testing to take place during a single appointment in a primary care setting. A clinician could collect a saliva sample quickly and comfortably, without specialist equipment, and without referring the patient for invasive procedures unless necessary. This shift would represent a major change in how early cancer detection is delivered. Instead of relying on referral-based specialist diagnostics, early screening could become part of routine care. This has the potential to reduce delays, lower anxiety, and improve outcomes by identifying disease at an earlier, more treatable stage. It is important to be clear about the current stage of this research. This technology is still in development. It is not yet a clinical diagnostic tool, and it is not available for use in healthcare settings. Scientists are continuing to refine the hydrogel, improve the speed of protein capture, and ensure that the test can reliably distinguish cancer-related proteins from the many others naturally present in saliva. These challenges are expected in early-stage innovation. Addressing them carefully is essential to ensuring that any future diagnostic tool is accurate, safe, and trustworthy. What makes this research particularly significant is not just the technology itself, but the broader approach it represents. It reflects a move toward patient-centered diagnostics. Rather than expecting people to adapt to uncomfortable or intimidating testing procedures, the technology is designed around ease, familiarity, and dignity. There is also wider significance beyond oral cancer. The same hydrogel technology could potentially be adapted to detect other diseases, using saliva or different bodily fluids. This opens the possibility of broader screening tools that are non-invasive and accessible. From a public health perspective, early detection has far-reaching implications. Identifying cancer earlier can reduce the intensity of treatment, improve survival rates, and reduce long-term healthcare costs. It can also support people in remaining active, independent, and economically engaged for longer. This is where the concept of lives and livelihoods becomes particularly relevant. Improved early diagnosis does not only benefit individual patients, it reduces pressure on healthcare systems, lowers the economic impact of long-term illness, and supports wider societal well-being. From an educational standpoint, this research provides a powerful example of interdisciplinary science in action. It brings together materials science, molecular biology, clinical need, and an understanding of patient behavior and experience. It also demonstrates how innovation often emerges from rethinking familiar objects. A lollipop is an everyday item, but when combined with scientific insight, it becomes a diagnostic tool with the potential to change care pathways. For learners and professionals, there is a clear lesson here. Effective health innovation is not only about advanced machines or complex treatments. It is about asking better questions, reducing barriers, and designing systems that people are willing and able to use. It also highlights the importance of early career researchers and public investment in exploratory science. Many transformative ideas begin as high-risk concepts that require time, funding, and careful development before their full value becomes clear. As this research progresses, it will continue to be shaped by scientific evidence, regulatory oversight, and ethical considerations. Clear communication at this stage helps ensure that innovation develops responsibly and earns public trust. In that sense, this work is not just about detecting cancer. It is about reimagining how healthcare can be proactive rather than reactive, and how technology can be used to support earlier, kinder, and more effective intervention. Thank you for listening to Unicate Talks, a podcast for Unicate College. In today's episode, we explored how a deceptively simple idea, grounded in sophisticated science, could transform early cancer detection and improve both lives and livelihoods. And a special thank you to Dr. Watkis for his expert insight and guidance in shaping today's discussion, due to his background in public health and epidemiology. Until next time, stay informed, stay thoughtful, and stay curious about the science shaping our future.